Prepare to experience the strongest radio allowable by law. Secrets will be revealed. Myths dispelled. From the studio gym where excuses never apply, it's Superhuman Radio with your host, Carl Lenore.
Hey, welcome back to another episode of Superhuman Radio. Today is a special day. Today is November 19th, 2021. On this date in 2005, this show had its maiden voyage, its premiere episode. Today marks our 16th year in continuous production. Today's show number is 2,792. Wow, that's a lot of interviews. I feel like I've been in university for the past 16 years because I get to talk to the smartest minds in a variety of scientific spaces. And, you know, you have these conversations 3, 4, 5 days a week. It's like being in school. You learn things. You learn a lot of stuff. I have to tell you something funny. So Natalie Kapitsky, who is my social media manager and also helps me produce the show, she's in the background right now. Today, somebody walks into my office with a bag of Wendy's and 2 triple stack hamburgers. And I told a story. I was interviewed once about how the show got started and how I was 330 pounds and how my daily routine when I was running my alarm company was to drive through Wendy's. And this is when they had the double stack. It was a 99-cent 2-patty hamburger. I would order 6 patties on 1 bun. And the girl would always say to me, you want, I said, yeah, just throw the buns away for the other 2 sandwiches, put them all on one. And I used to do that 2 or 3 times a day with Biggie fries and a Dr. Pepper. And that's how I had, I did that every day consistently for years. And that's how I got to be 330 pounds and end up with a heart dysrhythmia. And so this guy walks in today from DoorDash and he's asking for Natalie. And I'm like, I don't know, maybe go up the hall and ask up there. And so finally he comes back. He says, is this Suite 103? I says, yeah. He goes, no, I think this is for you. I says, it's Wendy's. What the hell? I open it up and it's 2 of these giant burger patties with all the meat on them. And Natalie sent that for the anniversary show. So that was very thoughtful of her. Thank you. And I already ate one. I'll eat the other one after the show is over. Okay. So before we get started, we have to thank our title sponsor, and that's Legendary Foods, makers of the Tasty Pastry. The Tasty Pastry started out as an alternative to a Pop-Tart. It's become so much more with 20 grams of high-quality protein, less than 1 gram of sugar. For those of you who are carb conscious, 4 to 5 net carbs. But even though they're so good for you, they taste amazing. Everybody loves them. You will really believe you're eating cake. It's not like, oh, well, I'm happy it's 20 grams of protein and less than 1 gram of sugar. It kind of tastes okay. No, these things taste amazing. Go to shrnetwork.biz/legendary. Use the code SHR10 to save 10% off your order today. Check them out. And everything that Legendary Foods has is wonderful. Now, without further delay, bring my guest on. So we're talking today with Franziska Graef. She is a PhD candidate at I forgot what university. You're in Vancouver, right? What university?
Yes, UBC, University of British Columbia.
Right, and you work actually in the pediatric division, don't you?
That's correct. Yeah, I work in the pediatric division, but we do a lot of research that pertains to not just children, but also just basic research.
So this recent study you published is fascinating. I'm going to synopsize. Can fasting modulate infectious disease? It's a much longer title than that, but we have to make these titles fit YouTube's requirements. So, why this study? What led to this? What previous work that either you or colleagues or both were involved in that made this study necessary?
Yeah, so I came to Vancouver because I was interested in studying the gut and the gut microbiome, and this was about 7 years ago. And there wasn't anything out there yet on fasting and the microbiome at that time. And I had been interested in fasting for a long time. So I had proposed to my two bosses at UBC, like, hey, why don't we look at that? Nobody has looked at that yet. And they were, very hesitant, to say the least. And one of the reasons is, as you mentioned, because we work in a pediatric research environment. And so fasting and kids, is of course a big, you could say a no-go at this point. And he said, well, I don't know about, fasting, and, we look at IBD and inflammatory bowel disease and such. But he also has a research program focusing on infection. And he said, my first thought when I hear fasting is, that's got to be a problem when you have an infection. Like, you need to be strong, you need to eat. When you're sick, this this can't be good, and we can't recommend something like that to people and kids that are sick a lot, that deal with infections a lot. So I said, yeah, fair enough. Let me see what's out there yet. And I came across a study from 1979 and 1980, where a couple of researchers did something really interesting. It's actually two studies. So they infected mice with a lethal dose of Listeria, which is, an intracellular pathogen. And they did two things. One thing is they fasted the mice before, a group of mice before they infected them, and with a lethal dose, or known to be lethal dose. And most of those mice survived, surprisingly. And then they did another study where you may have heard about this, or this from experience. When you're sick, you often lose your appetite, right?
Mm-hmm.
You don't want to eat anymore.
Well, and there's mothers always used to say, you feed a cold and starve a fever. And I would say a fever is more often accompanied as a result of an infection, where the body temperature is rising to try to eradicate. P450 enzyme cascades speed up, and the body is trying to get rid of the virus. So we instinctively, even when I worked as a kid with horses, if a horse didn't eat, we thought, oh, he must be sick.
Yes, exactly. This is exactly right. So this is a whole set of what we call sickness behaviors. And as you mentioned, fever is one of them. Lethargy is another. Animals, if your dog is sick, the dog will retreat, will go into his little place and try to sleep, stay away from humans and other animals. Social isolation and distancing, is an intuitive response to that as well.
Interesting.
And another response is loss of appetite, right? And it isn't, or it still isn't, but hasn't been clear so much, whether this response is protective or not, And as you know, like with animals, we're more likely to let them follow this response, maybe. But grandmothers, when their grandkids are sick, they want them to eat.
Yeah.
Yes, eat when you're sick. You have to eat at least something, And anyways, in this study, the second part of the study, what they did is they force-fed the mice that were infected with this lethal dose of Listeria, and they didn't allow for them to follow their natural fasting response. And this then led to the fact that those force-fed mice died, more of them died, or they died sooner. And so I thought, wow, this is really fascinating, And this was sort of the only, study that I could find on that. And I said, well, why don't we look at that with regard to gut pathogens? Because the gut is also kind of the first organ that is really affected by fasting, if you think about it. And also nobody knows what's happening to the whole microbes, that are in the gut. And so this is sort of the background and why my boss then decided to give me a chance with the fasting and say, okay, sure, we have a bunch of models in the lab. Why don't you, pick a couple and try the fasting and see what you find? So
how was your study designed?
So I picked at first a Salmonella model because this model is a very common model in gut infectious research. It's very well known. It's very well defined. We have a lot of tools to look at all the different responses on the bacterial side, but also on the host side. And it's also very reliable. So every time, in this model that you infect the mice with this pathogen, they reliably get sick. There's not a lot of variation often there. So this is good to have. And so what I wanted to know too, is because I'm interested in prolonged fasting. We can talk about this a little bit more later.
We're going to talk about that later because we have some interesting stuff to talk about there. Go ahead.
Right. One of the concerns is when you're fasting, like if you're fasted or in a fasted state, if you're more susceptible to infection then, because you don't have the nutrients and maybe your immune system needs all these nutrients.
Yeah, you're weakened. Yeah, right.
Exactly, you're weakened. So the way we designed the experiment was to fast the mice at first so that they would be in a fasted state. And of course, we always have a control group that would be fed on the other side. And then infect the mice orally and see, what happens. And and what we saw, and this was completely surprising to us, was when I, fasted the mice, and this is for 24 hours, and then I infected them with a dose of Salmonella. The mice didn't really become infected, or at least we couldn't see any signs of infection.
No symptoms. Yeah, no symptoms of infection.
No symptoms of infection.
Right.
And this was completely, from my boss and everybody working in the lab, completely unheard of. Like, actually, the first time that I ran this experiment, I thought I had made a mistake. I thought,
right.
Oh man, I
did something wrong because none of them are getting sick, right?
Yeah, I thought for sure, I had mixed up, either the cages of the mice, or maybe I had forgotten to infect them, or like something weird was happening.
Yeah.
So I was like, okay, yeah, of course I got to repeat this, and then we repeated it over and over and over again. And all the time we would find the same thing. The mice that were previously fasted and then infected, they did not show any signs of gut infection. There was no damage. There was no, any kind of what we could see.
Is there a couple questions. So first of all, do rodents produce ketones? Do they go into a state of ketosis when they are not fed?
They do.
Okay.
And here is one of the big differences, and something I really want to stress. So rodents have a much, much higher metabolic rate than humans, right? And so a 24-hour fast may not sound, that long, but for a mouse, it is actually pretty long. It's a prolonged fast, and it translates to roughly, a week, 5 days to a week-long fast in humans. And this is something that is very often, misinterpreted when these studies, because most studies, let's be honest, about fasting, and especially basic research studies, are done in mice, And so you cannot translate this in terms of the timing of the fast one-to-one to humans,
Right.
So the mice were, they were in a profound fasted state. They had elevated ketones. I checked for that. Very low blood glucose. And when they were in this robust fasted state, this is when we infected them. And then they continue to fast.
Do rodents exhibit signs of hunger? Like, do they have, do they, for instance, I know lordosis, is a sign that the rodents want to mate. Are there signs that, oh my God, these rodents are very, very hungry? Or do they reach a point like humans do? When I used to do long-term fasts, once my ketone levels were up to like 2.5 millimoles, I had no appetite and I felt almost euphoric. Does that happen to rodents, or do they continue to feel hungry?
Yeah, so I mean the time frames are just much shorter for rodents, because already after 4 hours of a fast in rodents, their ketones start to go up slowly. So by the time you reach 24 to 30 hours, they will show some food-seeking behavior. So they will go to the edge of the cage, and they will just show food-seeking behavior for sure.
Okay.
And 48 hours is generally considered the longest that you can safely fast a rodent. Anything beyond that is not considered safe anymore for rodent.
Okay. So, so then you introduced food to them after the 24-hour fast, correct?
No, then I infected them after the 24-hour fast. I infected them, and so they were already in a robust fasted state, and they continued to fast for another 24 hours.
So so that's the longest anyway. So in that second 24-hour fast, they displayed no sign of the infection whatsoever.
Exactly.
Okay.
Exactly. And
but
even
then you fed them.
Then that was part of a lot of the times we stopped it right there and we just looked at the mice. Then, but for part of the experiment, we also refed them. And we compared this then to the mice that were fed throughout the entire time, right? And then the infection, started picking back up, but more slowly. And the damage that we could observe in the gut was also much more attenuated in the refed mice.
As opposed to the mice that ate continuously throughout the infection period.
Exactly. Their gut, their cecum, this part of the gut where this infection is prominent, was almost completely destroyed.
Yeah. And this happens to people too. So okay, so once you reintroduced food, they did show signs of infection, but it wasn't as bad as the ones that had eaten all along.
Exactly. Okay. Yes.
Now, did you test for any other biomarkers? Did you look at things like insulin, or could you look at levels of autophagy? I guess my question is this: do you think the protective effect is a result of some of the pathogenic microbes sporing because they were starving? Because we know that a lot of microbes don't die, they spore and they go into suspended animation. And once you start eating the food that they like, they wake up again and they start contributing to the chemicals that they make. So do you think that this was a result of the microbiome shifting because of some of these sporing? Or do you think this was the result of a conservation of resources, right? So the body has to mount a fight. The immune system has to mount a fight against the virus. But if you're eating too at the same time, the immune system is part of digestion. So now you're saying, well, here, give me half of those troops and put them over here so we can digest food. Or was it the result of autophagy being stimulated, which we know is stimulated when you don't eat? All three of those. Could you tell?
Yeah, we could tell a little bit. I can tell you, what we found. So the first thing that you mentioned, it seems to be the case that the competition of the nutrients between the pathogen and the resident microbiota seem to have played a big role, because what we found is the following. So first of all, so Salmonella and other pathogens, they're really cool in that how they infect cells is they have to, they form a little needle complex. You can imagine this like an actual injection needle. They make this, and then they inject toxins into the gut cell, and then the gut cell, is manipulated and engulfs the pathogen, and then, infection gets worse and worse from there. So for the bacteria to make this, to infect the host, they need a lot of energy to make this to build this little needle complex. And if they cannot build this needle complex, they are kind of like a toothless tiger. They cannot really they hang out in the gut, but they they can't really get inside the cell. They cannot really get inside the host. And so what we found is normally Salmonella, they go and invade the gut cells. But in the normal mice, when we looked at exactly where the Salmonella were located, they couldn't make it inside the cell. So we were like, oh, this is interesting. Normally, this is the normal response. The cells, the Salmonella, they always go and invade these cells, and then they cause all this damage from there. They couldn't do this. So we said, we wonder if, maybe the microbiome that is there, takes up either all the nutrients, or it's because of the shift of the microbiome to a different composition, that the host is protected. And so we use germ-free mice. And so these mice, they lack a microbiome. They've been bred to, be raised completely without any bugs. And we infected those mice and did the same protocol. And indeed, in these mice, the Salmonella, all of a sudden, we found them inside the cells, they could invade. So they were able to, have enough energy to form this little needle and get inside the cell. So this is, yes, part of the protective effect is, yes, the Salmonella were less, we call this virulent or virulent. They couldn't really infect the cells, and this had to do with the microbiome being there.
Shifted.
What we don't know is, we didn't show specifically, is this due to like a specific microbe being there, because the fast shifted the microbiome, and therefore, there's a certain microbe now there that wasn't there before, or
is there a microbe that was active before that's gone into suspended animation and is not contributing to the infection any longer?
Exactly. Or is it also nutrient competition, and the microbiome is just more efficient at taking away the nutrients?
See, I don't think it's nutrient competition. I'll tell you why, because I've learned that the worst microbes seem to be the strongest and most robust. And so they tend to be the most competitive. Yeah, they're the hardest to kill. They're the ones that gobble up all of the nutrients that all of the resident microbes need. They're just, they just overperform. And so I don't think that was it. Yeah.
Yeah. I mean, it's possible. I mean, we, I can tell you that we did some follow-up studies too, where we tried to, and this is not published yet, and we're still working on this.
Okay.
Where we tried to reintroduce single nutrients, just glucose and just this and just that, to see if this would be enough to get the bacteria up and running again. But we didn't find yet anything like any specific.
Did you use flour?
Cornstarch.
Okay,
cornstarch. We used cornstarch.
Acellular carbohydrates. We did a show like 10 years ago. Acellular carbohydrates, anything that has been ground up into a flour, feeds the pathogenic microbes in the stomach before it even goes downstream. And so that's why we need to stay away from acellular carbohydrates, powdered starches in general. Yeah, they're good points. You gave them that. Yeah.
Interesting. Yeah, I didn't know about that one. Yeah, and so, but to go back to the germ-free mice, those germ-free mice that were fasted and infected, the bacteria could go inside the cell, and they could cause a little bit of inflammation, but still, nothing compared to the germ-free mice too that were fed throughout the infection.
Yeah.
And when we looked at certain markers, for example, maybe you have heard of NF-kappa B, it's like a central regulator of inflammation.
Inflammation, right? Yeah.
Yeah. It was completely like not expressed, on the protein level in the fasted mice. And so this is an interesting and a more recent concept, that is called disease tolerance. So there's disease resistance and disease tolerance. I'm not sure if you have discussed this before, or
no, we haven't. I mean, I get the difference, but please, excuse me.
Yeah. So disease resistance is kind of what we think about generally when we think about the immune system, right? The immune system is there to kill pathogens,
and it's robust,
it's there to reduce their numbers, to kill them with any means possible. In the process of doing so, a lot of the time, though, the host or the body itself also suffers. You have, neutrophils like making oxidative burst, and so it kills the bacteria, but it also damages your own tissues. And so this is one part. And then the other part is disease tolerance, where if the host can not do this, but tolerate the bacteria or the virus or whatever being there without, causing this immunopathology, this can also be beneficial. And so there needs to be a fine balance between this tolerance and this resistance. And so what we found is fasting seems to increase this tolerance, from the host side. So you just don't react as strongly to, let's say, the Salmonella or other bacteria, which, can be beneficial because pathogens like Salmonella, they exploit inflammation. They love it if there is inflammation, if there's immune cells, if they spew out all these reactive oxygen species and all this stuff. Salmonella thrive of that. And so if the host does not do that and is more tolerant and just tolerates the bacteria being there and doesn't respond with a full-fledged immune response, they become also less virulent and you have less damage. But of course, there always needs to be a balance, because as you can imagine, you cannot let that run forever, because at some point the body needs to take care of the pathogen.
Right. What about biofilms? Does the body produce biofilms when fasting? I first learned about biofilms doing a show about human breast milk, and mothers in the 90s that had HIV who breastfed their babies, the virus was sequestered in biofilms and the baby did not get infected from the breast milk. And so biofilms seem to be this duct tape of the stomach. When it has to fix something, just wrap it in biofilms and let's get it out of here, and it'll be safe. It's like a virus vault. Does the body use biofilms when we're fasting?
That's a very good question, and we don't know yet. But what we observed is that the bacteria in the gut thrive that live off host-produced factors. So, for example, this one species, Akkermansia muciniphilia, this is a species that lives off host-produced mucus. And this bacteria thrives during fasting. And we suspect it's because there is no other nutrients there. And so it turns and it feeds off the nutrients that the host provides. And we suspect that this can lead to something like a biofilm formation along the epithelial cells, because this is where the mucus is found. But we haven't shown this, or nobody has shown this yet. So we don't know yet if fasting induces biofilm formation.
Interesting. Akkermansia is fast becoming a very popular probiotic. We have companies now selling it. It's seen in fasting. It's also it's diminished in the carnivore diet. People who just eat meat, it seems to disappear.
Yeah.
But I never knew that it's fed on mucus, which is fascinating, because mucus in the gut, which is not the same thing as the mucus in your nose, protects the lining of the stomach.
Exactly.
So,
it's very important.
Akkermansia is present when there's adequate mucus in the stomach, which means that the stomach must be fairly healthy in order for Akkermansia to occupy that space.
Exactly. Yeah, this is exactly right. And with Akkermansia, it's fascinating. You're right. It's popping up left and right in all these studies. And there's also some mouse studies that show sort of detrimental effects, that all of a sudden, if you have only or a lot of Akkermansia that eats up all of this mucus, you become more susceptible to other infections.
Irritable bowel syndrome starts out with the mucus disappearing. So it's interesting because as humans, we're like, oh, Akkermansia, I want more, and it's like it's all about the balance.
It's yes, exactly. It's all about the balance. Yeah,
the gut is a democracy. And when it comes out of balance, you have chaos,
Exactly.
We're going to take our first commercial break. We have lots more to talk about. You are watching and listening to Superhuman Radio. Stay tuned. We will be right back. Being superhuman means being conscientious about the choices we make. White Oak Pastures is superhuman when it comes to raising livestock. They're committed to animal welfare, land regeneration, and rural revival. They responsibly manage the entire process from the pasture to your doorstep. High-quality, pasture-raised, great-tasting meat from a farm you can trust. Full nose-to-tail selections. They even have options for your pet. Go to shrnetwork.biz/whiteoak and use the code SUPERHUMAN to save 10% off your entire first order.
New Mass Pro Synthogen X2 just upped its own legendary game. To distance itself even further from the rest of the pack, Synthogen X2 now has double the key active ingredients. If you've ever wondered what steroid-like recovery feels like, Synthogen X2 delivers. See why others compare it favorably to powerful bodybuilding drugs at synthogen.com. Mass Pro Synthogen. When you train with it, you'll gain with it.
I would easily say that I am the hugest proponent you will ever meet to doing anything that will improve the quality of my sleep. And that's because sleep is linked to just about every metabolic disorder we see in our population today. One of the easiest things you can do to improve the quality of your sleep is to get a pillow that can be shaped into the exact form factor that allows you to get your best night's sleep. And that is MyPillow. I've been sleeping with MyPillow for a few years now, and I can tell you that when I have to travel and stay in hotels, I don't get a good night's sleep because I don't have MyPillow with me. Right now, you can save up to 60% off of everything offered to improve the quality of your sleep at shrnetwork.biz/mypillow when you use the code SHR. Or you can call toll-free 800-889-4938. And remember to use code SHR to save up to 60% off of everything at their website. You already know that Magnesium Breakthrough is the single best magnesium supplement in the world and quite possibly the most popular right now. So Bioptimizers doesn't have to do anything special to get your business, but they are. Black Friday starts now and runs the entire month of November. They're also giving away over $200 worth of free gifts. Bioptimizers is a legacy advertiser on this show and gives us great support to keep bringing you great content. And now they're rewarding our audience directly. Throughout the entire month of November, go to magbreakthrough.com/SHR and use code SHR10, not only to get a great discount, but to get $200 worth of free gifts as well. And while we all know that Magnesium Breakthrough is the single best magnesium supplement in the world, Bioptimizers stands behind all their products with a full refund up to 1 year after purchase. No questions asked. Go to magbreakthrough.com/SHR and use code SHR10 today. Don't waste another minute. Have you been struggling to achieve results in the gym? Trouble sleeping through the night? Overwhelmed with stress or anxiety? Look no further than Infiniwell supplements. They have designed products to address specific needs like muscle growth, hormone balance, sleep, and anxiety from the first use. Infiniwell helps you live well. Go to shrnetwork.biz/bewell, that's BEWELL, and use code SHR20 and start living well today.
This is the Superhuman Channel. Evolution just got kicked up a notch.
Welcome back. We're talking with Franziska Graef, and she is a PhD candidate. And we're talking about how fasting may actually benefit us when we are infected. So this word infected for a second. We have lots of viruses that we get sick from. We have just gone through a pandemic and still in some remnant of that at this point in time. We are talking about in this case, the study, opportunistic viruses that seem to attack the gut. The immune system is the gut. So when we talk about immune system function, robust immune system, we're talking about a healthy gut. The inflammation is the army of the immune system sent out to get rid of bad actors. What about viruses that don't attack the gut? Maybe like this current pandemic, they seem to attack the lungs. Is there any reason to think that fasting would benefit just about any type of viral infection?
Yeah, that is an excellent question. And I mean, the true and quick answer is we don't know that yet. But there is some pointers maybe that, we have. There is, in mouse studies, what they have found with different viruses, for example, influenza A virus, was that fasting was more detrimental in that case. So when the mice were given glucose, they actually, and a dose of influenza, they actually survived more. But then there's also, I think, a study where mice were put on a ketogenic diet and given, I think, also influenza A virus, and those mice did better. And so, and there's a couple of other studies, and they all show, different kind of slightly different and opposing effects. And so it's not quite clear at this point, when and how, fasting during viral infection is beneficial and when it's detrimental. But, the fact that, for example, with this current pandemic, people that have diabetes are more susceptible to severe forms
symptoms.
of,
yeah. In fact, we covered a study that showed that patients with mild to moderate symptoms, when brought into the hospital, who had the highest blood sugar levels, their symptoms got worse faster and they died. And the study shows that there was a direct link between blood sugar management, blood sugar levels, and dying from that virus. So it's interesting.
Yes, exactly. And so this is where I think, this could play a role, but it's hard to know at this point, because there is a fine balance again between your immune system responding and needing to respond to get rid of the virus, and you kind of dampening the immune system, because that has also been shown with this virus, right? That sometimes people die from an overactive immune system. All of a sudden, if it
cytokine storm.
Exactly. Full-blown cytokine storm, you also have a problem. And then it's also interesting, I was involved in a little project. This data is also not published yet, but also with SARS-CoV-2. And this was just in vitro, so just in cells. And from that experiment, it wasn't quite clear whether it's the high glucose that could maybe lead to, faster viral replication, or maybe the insulin. So, but of course, those are very tightly linked. So if you have high glucose, you're mostly bound to have also higher insulin levels, or maybe you're, insulin resistant or something like that. And so that definitely plays a role in. There's
another aspect to this that I'd like to share with you, if you've not heard of it before. Dr. Mikhail Blagasclony was a frequent guest on the show, but he's kind of taken himself out of the mainstream right now. And he is the oncologist turned anti-aging doctor who promotes the use of rapamycin as an anti-aging. I take 6 milligrams of rapamycin every 2 weeks. Rapamycin reduces the presence of senescent cells. Senescent cells are the cells that this SARS-CoV-2 occupy to become virus factories to replicate. Senescent cells possess a higher level of reverse transcriptase, and reverse transcriptase with these retroviruses, it turns those cells into factories. Now this is anecdotal, but there is a lot of good research to support this from Blagasclony's group. If you remember when we first had this pandemic start, there was a cruise ship that was sequestered out at sea and not allowed to come back because there were so many sick patients. There was a husband and wife, and they both had COVID. The husband got better that day. He ran a mild fever, and he was released and flown off the boat the next day. The wife stayed for 4 weeks, or whatever the length of time they were out in the ocean. He was the recipient of a kidney transplant, and he was taking sirolimus daily, high doses, to ward off organ rejection. Well, sirolimus is rapamycin, and these things reduce the viral load by shutting down the factories that particular virus likes to occupy. There's one other example. Viruses are not intimidated by antibiotics, but this one. So zithromycin has been approved by the Food and Drug Administration to be used as an anti-aging drug, because one 5-day round of zithromycin reduces senescent cells in the human body by 97%. So one of the things you have to look at to fairly assess what's going on, and the other thing is, people with type 2 diabetes have the highest level of senescent cells. I did this show with a scientist from Rutgers University in 2007. Glucose signaling is what causes cellular senescence. So when you tie it all together, these people have ragingly uncontrolled blood sugar. They accumulate a lot of senescent cells. They age faster as a result. And they are more susceptible to the increased viral load because they have more factories available to be leased by this virus.
Yeah, this, it sounds plausible. I'm not sure about, the studies specifically. And, there's always the problem with anecdotal evidence as a scientist. It's definitely can give you some hints, but it's not, what we base our recommendations off. But, I agree. And, mTOR, target of rapamycin is modulated, of course, greatly by fasting.
Yes.
It's shut down.
AMPK goes up, mTOR comes down. And that's
exactly.
what rapamycin does. It does it for 3 days at a time. And so it's fascinating. But Dr. Michael Lasanti, who's in the UK, who's been on this show, he wrote a study showing that the reduction in senescent cells halts this virus. I had him on the show in 2020 at the onset of the pandemic. So there is good research out there that links senescent cells, the eradication of senescent cells, and the reduction in mortality in this virus. So it's fascinating.
That is fascinating. And, this is probably what I would say in general towards fasting and viruses. I would use it as a preventative strategy. So if you are, fasting, let's say how I like to fast is twice a year for, 1 to 2 weeks at a time. This has been shown to have a bunch of really good effects. As you say, yes, it does reduce senescent cells, it's anti-aging, it rejuvenates your immune system, all these different things, then you will be in a better state to respond to a virus. But I would caution at this moment, because we just don't know, to fast, let's say, if you actually suffer from the infection or from the virus, and to, just go on a full-blown fast. This is probably something that, at this point we cannot recommend, just because we don't know what that would do, if that would make the outcome better or worse.
But a lot of people fast when they get viruses. And I was going through a divorce and living in an apartment in 2008. I contracted swine flu.
Oh my God.
I was too sick. Luckily, it happened over a weekend. I was too sick to get out of bed and make anything, and nor did I have any appetite. So I literally laid in bed for 2.5 days, of which I got better. And then I came to work. I lost my voice. So I joked with everybody, oh, Barry White is doing the show today. But I came to work. But I fasted because of necessity. I couldn't get out of bed and cook for myself.
You know what? You're right. So I would rephrase it in that if you have this loss of appetite, which often comes with certain infections, bacterial and viral, I would also follow this natural response. I would not force feed myself if I lost my appetite upon infection. But again, this can only, this, in my opinion, only goes so far. So if you have just like with fever, it's okay to have fever for a few days, but if it goes up to a certain degree, or if you have fever for weeks at a time, this is no longer adaptive. This is probably maladaptive. And the same is true for fasting and losing this appetite. For a few days, this is probably really good, even when you're infected. But if it goes on for, longer, weeks at a time, and a high fever and such, then you probably need to do something.
Well, I want to make a supposition before we start talking about fasting in general. Those of us who fast are said to be metabolically flexible. We can go long periods of time without food and still function. The insulin-resistant model, they can't do that. If somebody has type 2 diabetes and they get a virus, and fasting is the best thing for them, but they don't lose their appetite, because every couple hours they have to eat, or they start to have tremor and sweat and anxiety. And so the lack of metabolic flexibility may be the decision, the pivot of whether fasting is going to be good for you or not in an infection circumstance.
Yes, I agree with that. And malnutrition, as people always, when I say, yeah, malnutrition, they always think about undernutrition, and it has been shown that if, like, let's say kids in Africa that have been really, undernourished for weeks or years, they are also more susceptible to viruses. But malnutrition is also, on the other hand, obesity and, having only been eating like a high fat diet and not having all the nutrients you need. This is also defined as malnutrition. And so if you're malnourished, one way or the other, either through, let's say, a Western diet or through really not having been able to get enough food, fasting, yes, you don't have this metabolic flexibility and you don't have the resources to properly make it through a fast. I think this is very true, and this can determine whether the fast will be beneficial or not.